Welcome to Vibe in Action, where we get to witness bold actions changing our lives and shaping the future.
Oh, it's wonderful to be here again today with another new face. Could you give us a brief introduction?
Hi, my name is Bon-Kyoung.
Welcome aboard, Dr. Koo. We all love discovering new things, and I can't wait to learn more. So, what is the topic that we will be discussing today?
It's going to be an innovative new technology in biology, which is called organoids.
Organoids. I've heard of something oid and something organ, but never together. Organoid.
Organize is a familiar term, but organoid.
Organoid.
I get annoyed.
What does that mean?
Organoid literally means something organ-like.
Like humanoid.
Yes. But this is organ-like, which means that we are growing small pieces of tissues in a dish, which resembles the substructure of an organ.
Wow. So you create these mini organ-like things separately?
Yes.
In vitro.
In vitro, which means in a dish.
Just
in a
randomly,
dish.
you get it to grow in a dish?
It is a sophisticated procedure, so you have to add on appropriate growth factors, etc., to really create a human body-like environment so that stem cells can grow into this 3D structure, which we termed as an organoid.
We'll get to the details later.
Wow.
We just heard only the basics, but it's already so fascinating. So before diving in further, shall we take a look at a short clip first?
Sounds good. Let's kick it off then, okay?
Vibe in action. I still marvel at how they do organ transplantation or blood transfusion, but creating an entirely new organ from scratch is on an entirely different level of technology, right? Yeah. You mentioned that people often call these organoids mini organs. Is it because they're normally size and they're made at a small scale?
Yeah, they are at a small scale, which is true. But in reality, they are not quite the level of organ yet. That's why we still call it as an organoid. In reality, I think the most precise description would be calling organoids just as an organ-like structure because they resemble only part of the organ at the moment.
So they're not actually small or miniature organs, but speaking more correctly, they are partial organs or pseudo-organs that mimic the structure and function of the organ?
Yes, so that's probably the right way for public to understand organoids. But I also need to mention that people working with organoids are trying to make it better and better so that eventually we hope that we can make it almost like the organ that we can actually utilize.
Oh, like in an identical manner. So when did people start making organoids?
That's not so long ago. Around the time I was doing my postdoctoral training in 2009, Dutch scientist Hans Clevers, who was also the mentor during my time in the Netherlands, he discovered how to grow adult stem cells into the 3D structure. He succeeded it firstly in the intestine, and then he continuously successful in other organs.
Wow, so you were with your mentor at the time of discovery?
Luckily, yes.
Wow.
I
What a moment.
witnessed all the new discoveries that were happening there.
On the cutting edge.
Yeah, you were there at the historic moment. And I love his name, Hans Clevers. And he cleverly developed the very first organoid, and it started off with an intestinal organoid, right? But 17 years later, what kinds of organs can be made?
Pretty much everything that you can imagine now. So
Really?
almost all the tissue that we can isolate from human body can be grown as an organoid. There are a few exceptions, such as heart and the brain, because they lack stem cells. And for those two organs, we have to utilize embryonic stem cells or pluripotent stem cells.
Which stem cell?
Pluripotent stem cells, which is equivalent to embryonic stem cells.
So that's different from the adult ones.
And at this stage, it is not possible to make the heart and the brain, not because they are too complex, but because they lack stem cells that are necessary.
Yeah, in adult body, we lack stem cells in the brain and the heart. For those two particular organs, we have to start from embryo stem cells.
And that's a different field, is it? Wow. It's hard to believe what you're saying, that we can create many different types of tissue already. But does this mean potentially we can cure incurable diseases or even infertility? And I wonder if that was the goal from the very beginning with your mentor.
I don't really think that was the actual goal in the very beginning, but he wanted to grow intestine in some way.
But was your mentor Victor Frankenstein?
Is that the goal?
That is so Mary Shelley.
That's not the goal, surely.
No, not exactly, but he wanted to simply grow the organ as a 3D structure.
Yeah.
And it was a very accidental finding.
Wow, really?
I want to quote something here. The most exciting phrase to hear in science, the one that heralds new discoveries, is not eureka, but that's funny, said Isaac Asimov.
Oh, because lots of discoveries come by chance,
Yeah,
right?
by
Unexpectedly.
accident.
Wow.
Create a body-like environment.
It's easier said than done. I mean, it's not the idea, but the execution that matters. So what exactly does it mean to recreate an environment similar to the human body? How do you do that? Please tell us step by step.
First of all, we know how organs are maintained in the body because of the stem cell activity we have in each organs. And they are relying on growth factors, which we call as a niche signaling. And by creating this particular signaling environment in a dish, we can allow stem cell just to grow as if they were in the body.
Wow. You're kind of tricking them,
Yeah.
saying you're in the body.
You're making it sound so easy.
Yeah, you just give them the growth factors, there you go.
Let's say the organoids or the tissue cultures are basically derived from stem cells, and these cultures can be crafted to even replicate other human organs like the lungs or the brain. But how do you prompt or instruct those cells to progress through appropriate steps to generate properly functional organs?
Depending on the type of stem cells you work with, we are taking different approaches. For example, for the adult stem cells, you just need to provide the tissue environment in a dish, then they know how to grow the particular organ.
Wow, they just do it.
Yeah. But in the case of the embryonic stem cells, you have to tell them where to go by changing the media with the different signals so that they can eventually make either heart or the lung or the brain, depending on the growth factor combinations.
So what is the signal? Is it like electric signal or verbal signal or light signal? What kind of signal is it?
Yeah, so in biology, signal could mean many different things, but they are usually utilizing proteins to communicate to each other.
Proteins.
So cells getting different proteins to sense and then they interpret the protein combinations to do the exact order that they have to follow.
So the final destination, the destiny is not predetermined or pre-written in the seed itself, which is the stem cell, but you can actually talk it into becoming something by giving directions.
That's what the development is taking place. So depending on the region of the body in the developing fetus, cells getting different signals, and they determines what they become at the end.
Based on their
You're
environment.
making me visualize scientists in a row, like putting a spell on a bowl of stem cell and like wielding a magic wand saying Wingardium Leviosa. You say, grow into a kidney, grow into a lung.
And voila,
Yeah.
you've done it. Like you said, it might not look like or be 100% identical to the organ, the organoid. So how do you test if it works? Do scientists like implant it somewhere into an animal or something like that?
Oh yeah, some people have tried to put the mouse or human organoids back to the mouse brain or some different part of the mouse body.
Yeah.
That's one way of testing it. But we can also test various biological functions of the particular organ, all in a dish. So in that way, we can confirm that they indeed have the designated functions that we wanted to have from the organoids
Wow, in the lab.
in the lab.
I know you've oversimplified things for us to understand, but frankly, it's almost like witchcraft. It's so hard to believe. But there are things that we cannot verify with eyes. For example, there could be different kinds of pain originating from stomach.
Yeah.
Sometimes you feel nausea, sometimes you feel like gnawing sensations, or it can be crampy. But you can't really tell or differentiate the feelings by the look of it. How do you know that it's really working and it's really properly made?
Yeah, if there's no like chemical function or output, right?
Yeah.
And especially with the brain, where so many things are going on, it's so complex,
And
right?
a specific organ is connected with brain, so it sends off signals off to the brain, and sometimes it receives signals from the brain. The connection goes both ways. So how do you know for sure that it's working?
Yes, we biologists would also like to hear the voices of the organoids.
Yes.
Because they can then just tell us
Are you
whether
well?
everything is all right or not. But the way how we do is we understand the gene expression as the response of the organoid. So by comparing the gene expression patterns of organoids to the actual organs, we can tell whether they are 100% resembling of the target organ or not. There are also many other assays with which we can tell whether they have a correct function, as we assume.
But I think the intestine is relatively simple. What if it's other organs like the eyes? How can you tell that it's actually seeing the object?
I'm not sure whether we can actually make them see something, but by providing some electronic signal or light, we can measure whether the cell sensing those with some response. So the neuronal activity can be measured from the organoid to really say whether they actually get a response from the input.
Wow. So you can't be sure that it is working, but you can at least be sure that it is responding.
Yeah.
Wow, so amazing. Is it safe to say that with the current technology, we've pretty much reached the stage where we're developing organoids that have been validated through all these tests?
Nowadays, people are trying to utilize organoids as a test platform, even for new drug development. So it is becoming more and more relevant to use organoids, even more than animal cases, because we can use human organoid directly without using any animals. So in that case, we can perform human experiments by using organoids that are grown in a dish.
Ethically sounds more sound as well. now I can understand how stem cells are created, right? And how you can also evaluate their quality. Does this mean they're being used in humans now as well?
Organoids can actually be used for transplantation, at least for now in animals. And there are many people who are trying to utilize organoids as a cell therapy, including some researchers in Korea.
Well, ideally speaking, it would be best if we could use these organoid models as true avatars for human organs that can be transplanted directly into the affected area. But it doesn't seem like quite there yet. So how far along are we on this journey?
Firstly, organoids, as we talked about the scale, they are very small compared to the actual organ size. So we cannot really replace a real organ with the small, tiny organoids. So what we are trying to do is a cell therapy, which means that we are utilizing cells from the organoids to refill the dysfunctioning organ in human body.
Like a patchwork.
It's a patchwork. But even for that, we have to follow GMP protocol, which is good manufacturing practice. And to achieve that, we have to change many things still to grow organoids at the highest quality so that we can utilize them in the human body.
And there surely will be such a long series of tests and trials, just like with drugs. It might be even longer than that, right? Many, many years or decades potentially.
I think it can be even more complicated than
So
just
not
a
in my
small
lifetime.
Wow. Oh, fingers crossed if we live to 150, though. But there must be concerns as well, especially maybe when you're putting the human organoids into animals. That's a bit not what nature intended, perhaps.
Right. It feels unsettling to imagine putting a human brain organoid into a mouse or a chimpanzee. And it raises the question of to cross or not to cross the ethical boundaries. There must be a lot of issues that need to be discussed thoroughly on top of the technological limitations.
So people often have a misunderstanding that a mouse with a human brain may have a human-like cognitive function.
You could imagine.
The manifestation of that is Mickey Mouse.
However, I think this is not quite the level. And stem cell biologists, including scientists working with organoids, they all follow very strict ethical procedure so that we don't really do something crazy. However, I also want to mention that ethicists and lawmakers, they should also work really hard because the progress of science is very rapid nowadays, so that the regulations and other things should follow as quickly as possible.
Yeah, we cannot just take the wait and see approach.
Yeah.
Well, ethical dilemmas and considerations are inevitable when we want to implement new technologies. And so it's important we stay conscious and responsible, but at the same time, we cannot just slow down the technological advancement, considering the immense benefits we can get from this innovative technology. Good news is organoids are already entering a stage of real-world application, and that you're continuing to take on some of the greatest challenges of our time. Shall we head over to your lab and see you in action with your sleeves rolled up?
Oh, sounds good. Okay, 5 in action.
Korea's heart of basic science, the Institute for Basic Science in Daejeon. At the Center for Genome Engineering here, research goes beyond cultivating organoids. They are tracking the life processes unfolding within them. Every time cells divide, mutations are created. Breaking away from flat, two-dimensional cell observation, researchers are now observing that very moment inside organoids, three-dimensional models that mimic human organs.
2차원 구조 세포 연구에서는 주로 세포 개별적인 행동에, 개별적인 반응에 조금 더 집중을 했었다면 오가노이드 수준의 연구에서는 여러 세포들이 모여 만든 유기적인 집합체로서 세포들 간의 상호작용과 행동에 대해서 조금 더 자세히 바라볼 수 있게 되었습니다.
The secret behind this tracking lies in fluorescent color. Using gene editing technology that precisely targets specific DNA, cells are labeled with different colors. If a region within an organoid becomes filled with a single color, it means that a mutant cell has pushed out its neighbors and taken over that territory.
We can study the behavior of mutant cells and wild type cells at the beginning, and we trace both of them together, and we can see the early mechanism of tumorigenesis.
One of the biggest limitations in cancer research has been that cancer could only be observed after it had already grown. Organoids have, for the first time, overcome that limitation.
The mutant organoid grow much faster and bigger than wild-type organoids. With the organoid system, we can do a lot of different experiments using this one. And then we see this is a real mechanism how oncogenic mutation actually hits and then communicate with the microenvironments and other cell types.
So-called mosaic genetics, which visualizes the seed of cancer. The European Research Council has recognized the exceptional value of this technology. Their research, now recognized on the global stage, is evolving beyond treating current diseases toward preemptively preventing future pandemics. The team has found clues in animals within natural ecosystems. Using zebrafish, known for their remarkable regenerative abilities, they are tracking the mechanisms of cellular recovery. They have also succeeded in building organoids from 5 species of bats native to Korea, key carriers of emerging viruses.
다양한 동물 유래 오가노이드를 가지고 있기 때문에 이런 동물로부터 올 수 있는 인수공통 전염병 바이러스들에 대해서 빠른 진단이 가능하게 되고 또 이런 실질적인 세포를 이용해서 병인 기전 연구가 가능할 수 있고 이럼으로써 우리가 어떠한 약제를 개발했을 때 가장 최적의 효능 검증이 될 수 있는 모델을 가지고 있고 이를 통해서 빠르고 신속한 항바이러스제 개발에 도움이 될 수 있을 거라고 생각을 하고 있습니다.
And now, the team is building an organoid zoo project, bringing the global ecosystem into the lab.
We basically establish adult stem cell-derived organoids from various animals. If you can think of zoo animals, you can think of elephants or tigers or whatever. And these are very difficult to research on a more genetic level, especially if we want to do genetic manipulation or study their genes. So having a model, like a 3D cell line, like organoids, is very beneficial to studying these type of aspects.
Organoids are emerging as the most complete platform to replace animal testing. The government has designated them as a key advanced biotechnology and expanded R&D funding to approximately $500 million. In this intense laboratory, organoids are evolving beyond tools for treatment into a powerful language that will protect the future of humanity and the ecosystem.
So we've moved beyond simply creating simple organoids, individual ones.
Which was not simple at all.
No, that's already tough. But we can now prepare not only for diseases, but maybe future pandemics. How do you see organoids and their application expanding in the future?
It's a very good question. And I think in the near future, what we are actually already doing now is we are putting organoid on a chip.
Chip?
Wow.
Like a processor?
What I mean by is the media and the organoids can be put on a chip in a regular size so that they can have a very standardized action upon any stimuli. So this can then be used in many practical assays in biology and also in industry.
So that's a orga chip?
Organ chip.
Orga chip?
We call it organoid on a chip.
Organoid on a chip. Wow, that's unbelievable.
And the second example that I know is that there are also people who are trying to put on electrode to the brain organoid so that we can connect brain organoid with a computer system so that we can, in some way, read what the brain organoid is actually thinking about. And we can also use the brain organoids to control some devices through the electrode by using computer system.
You're not writing a novel?
It does seem like sci-fi, doesn't it?
Yeah, and you also have a plan to establish an organoid bank?
We are planning to establish organoid bank from various animals. We call it Organoid Zoo.
Organoid zoo. Doesn't sound as fun as going to the zoo with the children, but still
It sounds
more
like a
meaningful.
cabinet of curiosities. There's no end to human imagination, and I have a feeling that I thought I would love lab diamonds only, but I have a feeling that I would love lab-grown organs as well in the future.
A new pair of eyes or something like that.
Yeah. And I'm adding quite a few new terms to my glossary today. Building an animal organoid bank, my gosh, this could be a game changer in the history of medicine, benefiting not only us humans, but also our animal friends. As a true pioneer in this organoid research, what is your ultimate goal, may I ask?
As you have mentioned, organoids have been very helpful for understanding human health and human disease. And I'm trying to extend it to all the other animals by creating organoid zoo. This will extend our capability of studying biology, not just in human-related issues, but also in those superpowers that we see from different animals who are living longer than us, who are also having higher regenerative activity than us.
Wow, what an ambitious concept.
Amazing.
Well, maybe in the faraway future, I'm gonna buy a set of fresh new organs
Yeah.
to my parents for their birthday.
Like the animal ones.
Yes. No,
Like a dog's
human
nose,
one, no.
eagle's eyes.
No, like happy birthday, mom. Here's your new lung. It's an organoid lung. That'll be awesome. Thank you very much, doctor, for your genuine contribution to the evolution of mankind. All of this has been truly encouraging and hopeful, and thank you so much for sharing with us. Hoping for the day when we overcome every disease and sickness on Earth. Shall we say it out loud one more time as a rep?
Let's do it.
Vibe in action.
Wow. So organoids future. That's like a movie, right? Robots inside the human body.
This is the future of the medicine.
Wow.
This should be one of the most powerful tools humanity will ever create.
Nanorobotics is kind of ultimate version of convergence of all those advancing technologies together.
What a medical wonder. There's absolutely no limit to technological advancement.